Okay, this is the discussion for the zonation in the Rocky Intertidal Lab. Up on the screen that you see right now would be some example data that you can use if you're absent. So take some time now to write down these data. The one column that is not included on this data table is the color code, but I included it with a name like acorn barnacles. I made blue, gooseneck barnacles were purple, etc. So you could just color in those colors, or you can actually even choose different colors if you want to use them. But these would be the data that you would put in that table. So go ahead and pause this video now if you would like to just leave this up on the screen so you can write that down. But for right now, I'm actually going to go ahead and we're going to scroll down to what the graph would look like. So if you did a stacked bar graph, this is what it should look like. Now I included data labels on mine to make it a little bit easier to see. You did not have to do that, although it might be helpful. But one of the things that we can see here is that as we go from the low tide line, which is 0 to 1 meters, to the high tide line, 8 to 9 meters, we see a change in the abundance of different organisms in each transect. This is a classic example of what we call zonation, or vertical zonation. So because of this, if you, for your hypothesis, said that biodiversity would change as we moved up from the low tide line to the high tide line, then that is supported, because clearly that's what we see here. Now let's go over the discussion questions for this. The first one, what organisms dominate the upper intertidal zone? Now the upper intertidal zone would include, let me go ahead and highlight it for you in yellow. The upper intertidal zone would include these three transects here that I'm circling in yellow. Okay, so that is the upper. So the organisms that dominate that would be the blue guys, acorn barnacles. Acorn barnacles have an adaptation that allows them to live high up on the tide line. The stress that they need to avoid here is desiccation, or drying out. So acorn barnacles can actually seal up their shells so that they don't lose water. Well, question number 2 asks what organisms dominate the middle intertidal zone? Well, the middle intertidal zone would be these guys. I'm going to highlight them in green. The transects in the middle. Okay, that would be what we could consider the mid-intertidal. And we can see here the dominant organism is sea sac algae. Now, what is the adaptation that allows them to do that? Well, just like in the upper intertidal zone, these guys have to deal with desiccation or drying out. So what algae actually can do is they can actually dehydrate or dry out to a crisp even, and then they just rehydrate later. They may also have a waterproofing material on their bodies that prevent water loss, that slow down this process. The third question is asking for the dominant organisms in the lower intertidal zone, and that would be the bottom three transects, the ones closest to the water. So the dominant organisms would be mussels, and we'll give an honorable mention to eelgrass as well. Now, the organisms down here have a much different stressor to deal with. What they need to deal with mostly in this case is not dehydration, but wave activity. These guys have to deal with waves. They don't want to be washed away. So mussels actually have byssal threads that they secrete from their shells that cement or stick them into rocks. And eelgrasses, being plants, have a very strong root system that keep them in place so they don't wash away. So that would be why these two types of organisms dominate the lower intertidal. Now the last discussion question, number 4, how does biodiversity change as you move up from the low tide line? Well, clearly it changes, maybe not necessarily in species abundance or species richness, but definitely in which organisms are dominating which zone. So the take-home point here is that different organisms dominate different zones because of the various adaptations they have for either wave impact or dealing with desiccation. So that wraps up our discussion for this lab. And I'll go ahead and stop the video. Again, you can pause it or replay it to make sure that you get the data and the proper answers for the discussion questions.